Okay, hello everyone. My name is April. I am the creator of these modules, and today I'm going to be walking through how to predict the cellular localization for your protogene. First, we're going to start off going to the Yeast Orphan Gene Project website, and once on the website, you can go to Lab Modules and select the Adopt a Protogene project. So I'll go ahead and open the localization guide and worksheet. From this Google Doc, you can go ahead and create a copy so that you can type on them. So the first tool is an online machine learning algorithm called DeepLoc. And how this works is it takes in an amino acid sequence and then it predicts the localization of that protein. So to get our amino acid sequence, we're first going to go to the ORF information website. And in the box, type in the ORF ID for your ORF. We want to get the sequence and then we'll click submit. So here is the amino acid sequence. Go ahead and click the copy AA sequence button. Okay, and then next we will go to DeepLoc's website. And here we'll paste in that amino acid sequence and then click submit. And so this can take a little while, just depending on how busy the servers are. I've timed it can take anywhere from 1 minute to about 3 minutes. So while this is loading, you can go back and look ahead at the questions. Okay, so here we have the results from DeepLoc. So this first table here is showing for each of the different localizations that your protogene can go to. And then below is the probability that your protogene goes to that location. So for example, it's predicting that for my protogene, there's a probability of 0.356 of it going to the cytoplasm. These numbers need to be compared to what the baseline for that location is. So what we would consider to be the background, and then how much is this probability over the background. And so by background, I mean, how likely is it for a protein in general to go to the cytoplasm? So to see what the result is compared to the background, there's this table up above called probability thresholds. So for cytoplasm, we see the background is 0.47. And for my protogene, it's showing a probability of 0.35, meaning it's below the background. This example specifically, the algorithm is not able to predict a localization. All of the probabilities are below the background threshold. Whenever the probability is above the background threshold, the box will be shaded different colors of green. So for example, the module example, it's showing here where it is predicted to leave the cell, so extracellular. And you can see the box is dark green because this probability is much higher than the background of 0.6. And it also predicts that for this example that it goes to the cytoplasm, but not as high probability. So for my example here, it does not go anywhere. And so you need to be careful. This up here, it says that it goes to the nucleus, but when we actually look at nucleus, it is not above the threshold. So when the algorithm is unable to make a prediction, it seems like here it just spits out a random compartment. So be careful when doing this. Okay, and then the graph below is showing which parts of the sequence were most important when making its prediction. For my example, it was not able to make a prediction. However, the things that the model focused on was this end part of the sequence. So the larger the letter, it shows more important the nucleotide was in determining potential localization. So next we're going to predict whether or not our protogene is located inside of a membrane. So whether it has any of these transmembrane helices or transmembrane domains. So to do this, we're going to go to TMHMM's website and we're going to paste in the amino acid sequence and click submit. This one is pretty fast and we have the results already. So here what we're looking at, what is the number of predicted transmembrane helices? So how many times does the protein pass through the membrane? Here it's predicting that there's one transmembrane helices. It contains 23 amino acids. So down below is a visual representation of these results, where the x-axis is the position along the protein. So each number is corresponding to a different residue. And the y-axis is the probability that that amino acid or that region is within any of these categories. So transmembrane means saying whether it's within the membrane. Inside is representing the probability that that region is inside of the lumen, so like inside of the ER or inside of the cell. And then outside is saying that's the part of the protein that is located like outside of the cell or outside of the ER. So if we just look at the purple line here, we see that there is a high probability in this region that these amino acid residues are within a membrane. And then this starts to taper off. And then if we look at the line that's representing the inside, so the probability that that region of the protein is either inside of the cell or inside of a organelle's lumen, depending on what membrane that this protein goes to. So if we look at the blue line, it's predicting a higher probability for the C-terminus of the protein to be inside of the lumen. Whereas when we look at outside, it's predicting that the N-terminal is more likely to be outside. So either in the cytoplasm or in the case of the cell membrane that is located extracellularly, so outside of the cell. So next we're going to use something called NucPred. This is an online algorithm that predicts the likelihood that your protein localizes to the nucleus. So we're going to go to NucPred's website, and we're going to want to go to the single protein link. And here again, we will put in the amino acid sequence. And here we have the output. So this first line here is showing the amino acid sequence for my protogene. Each amino acid is colored based on whether or not it is positively or negatively contributing to the sequence localizing to the nucleus. And the top line is showing the score for whether or not this sequence is likely to go to the nucleus. A score close to 0, like mine, means it is very unlikely, whereas a score of 1 means it's very highly likely that the sequence goes to the nucleus. In my case here, this protein is very unlikely to go to the nucleus. Pretty much all of the sequence is colored in this like kind of turquoise color here, meaning that there's really just no influence of any of these regions to suggest that this protein goes to the nucleus. For the sake of an example, I will show you one that has a higher confidence. So the score is still low when you consider that it can be out of 1, but for these protogenes, they tend to have lower scores in general. So a NucPred score of 0.3 is still suggestive that it could go to the nucleus. And here we see that there is a region in yellow. The nucleotides that are labeled in yellow have more of an influence in directing this protein to the nucleus. So we see here that there is a sequence motif that is suggestive that this protein can go to the nucleus, and it's this string of lysine residues here. Okay, so next there is another online tool that does something similar to what NucPred does, but this one's called TargetP. Like NucPred and the others, it's an online algorithm that takes in the amino acid sequence and then detects if there's any kind of sequence properties or sequence motifs that target the protein to the mitochondria or to the secretory pathway. Okay, so as before, we will go to the TargetP website, and then here we will paste the amino acid sequence of our protogene into the box and click submit, and this is also pretty fast. So here's the results from TargetP. It's in this table up here, and the column names are representing the different types of like sequence motifs. Other pretty much means it doesn't detect anything. Signal peptide is whether or not there are those sequence motifs that suggest the protein will go enter the secretory pathway, so go to the ER and then potentially be secreted out of the cell. And then if there's any mitochondrial transit peptide, so again, those motifs that direct the protein to the mitochondria. So the likelihood is representing the, how likely or how probable it is. So the values range from 0 to 1, where 0 means unlikely and 1 means very likely. And here, unfortunately, we see that other has the highest probability. So we don't find any of these sequence motifs. So it is unlikely that this protein is, I guess I should say it's not unlikely that these proteins go to the mitochondria or go to the ER and are secreted. It's just that it doesn't have the specific classical motifs that these algorithms use to predict. So the absence of a result doesn't necessarily mean that it doesn't go there. It just means that we can't be confident about it. Whereas if it has a high likelihood of finding either of these, then we can be more confident that it might go there and do follow-up experiments.